Okay, welcome everybody. This is the first part of the chapter dealing with the endocrine system. We want to talk about it as an adjunct to the nervous system in the maintenance of the body's homeostatic balance. It influences metabolic activity through hormones, which are chemicals that are secreted into the bloodstream. The response to the endocrine system is slower, but it's longer lasting than the nervous system, and that's due primarily to the fact that it takes more time to clear hormones out of the blood than it does to remove neurotransmitter from the synaptic cleft between the interface of a nerve and its target. Endocrinology is defined as the study of hormones in endocrine organs, how they control and integrate reproduction, growth and development, the maintenance of water, electrolyte, and nutrient balance in the blood, and how they control and integrate cellular metabolism and energy balance, and the mobilization of the body's defenses. When we look at glands, we can classify them into two broad categories. Exocrine glands, which produce non-hormonal substances, such as sweat glands and oil glands. They have ducts that carry secretions to a membrane surface or into the lumen of an organ, while endocrine glands generate hormones and lack ducts, and as a result they're generally highly vascularized because they have to put their product directly into the bloodstream. Endocrine glands include the pituitary, the thyroid, the parathyroid, the pineal, just to name a few. The hypothalamus is an example of a neuroendocrine organ and represents actually one of two places where the endocrine and the nervous system share structures, the other being the adrenal medulla. And this is the basis for a lot of claims of alternative medicine that utilize the manipulation of nerve endings or receptors in order to affect the body's overall health. The theory goes that if we can modulate what afferent pathways bring into the nervous system, we can affect how the brain controls the hypothalamus, how the hypothalamus controls the pituitary, and how the pituitary controls the rest of the endocrine system. Same thing is true with stress levels. The lower our perceived level of stress, the less innervation of the adrenal medulla, the less production of the hormone adrenaline, and the result is that the effects, the long-term effects on the body are reduced considerably. Some organs are both endocrine and exocrine in function. They include organs such as the testes and the pancreas and the placenta. If we take the placenta as an example, its exocrine function is to produce digestive enzymes that help us to chemically break down the macronutrients in our diet, while its endocrine function is to produce the hormones insulin and glucagon, which regulate our blood sugar levels. Other tissues and organs that produce hormones include fat, the thymus, the cells in the walls of the small intestine, the stomach, the kidneys, and the heart. And this is a look at the endocrine system. It's relatively straightforward. The pituitary gland sits right underneath the hypothalamus, which governs its function. The hypothalamus produces releasing factors that influence the secretion of anterior pituitary hormones. At the same time, the hypothalamus also produces the hormones that are secreted by the posterior pituitary. Basically, the releasing and inhibiting factors the hypothalamus produces will affect the production of what we call trophic hormones from the pituitary, anterior pituitary, which influence the activity of other endocrine organs downstream. While the hormones oxytocin and vasopressin, which are generated by the posterior pituitary, have as their effects, in the case of oxytocin, to control smooth muscle activity, which is important in responses such as birth and also antidiuretic hormone, another posterior pituitary hormone, which has the effect of causing the body to retain fluids and thus increase blood volume and blood pressure. On the back side of the brainstem, part of the midbrain, is the pineal gland. It produces a hormone called melatonin, and its job is to regulate our circadian rhythm, which is our sleep-wake cycle. The anterior pituitary produces trophic hormones, as well as hormones that control things like lactation and growth. The trophic hormones can target the thyroid, the adrenal cortex, and the gonads. And essentially, a job of a trophic hormone is to cause the target endocrine organ to produce its hormone. Most of these trophic hormones are regulated by monitoring hormone levels in the blood. At the level of the hypothalamus, this results in the production of either releasing or inhibiting factors in order to keep the levels of hormone in the blood constant. Downstream of the pituitary are the organs that are governed by hypothalamus and the pituitary to a large extent. This includes the thyroid gland, which produces the hormones thyroxine and T3, which increase your body's metabolism, increase the rate at which you burn fat, essentially, and raise your body's temperature. On the backside of the thyroid gland are the parathyroid glands. They produce a hormone called parathyroid hormone, which elevates blood calcium levels, and this hormone is an antagonist to another thyroid hormone called calcitonin, which tends to lower blood calcium levels. The thymus is essentially tissue that serves as a site of immunoeducation for the lymphocytes and the macrophages that help us fight disease, and the hormones it produces are thymosins and thymopoietins. Once these white blood cells have learned how to fight disease, they leave the thymus and populate other lymphatic organs throughout the body, at which point the thymus slowly begins to lose function and will involute and become scar tissue. The thymus sits in front of your heart and behind your sternum. Moving down, the adrenal gland is actually two glands in one. The adrenal medulla, as we learned in the chapter on the autonomic nervous system, is a neurosecretory tissue which is stimulated by the sympathetic division of the autonomic nervous system to generate adrenaline, which is dumped into the bloodstream and has the effect similar to the effect of sympathetic innervation, raising the respiratory and the heart rate, elevating blood glucose and blood oxygen levels, increasing the metabolic rate, dilating the pupils, and causing the body to retain more water, and also inhibiting peristalsis. While the adrenal cortex, which is the outer covering of the gland, produces steroid hormones. And the way I've always been taught to remember which area of the cortex does which job is to keep in your mind the mnemonic salt, sugar, sex. So what does this mean? What does this mnemonic mean? Well, the outer layer of the adrenal cortex, which is called the zona glomerulosa, produces the mineralocorticoid aldosterone, which controls your blood sodium levels by causing the kidneys to retain more sodium and excrete more potassium. So that's where the salt in the mnemonic comes from. The fasciculata, which is the middle layer of the cortex, produces glucocorticoids, cortisol and cortisone, which reduce inflammation and also elevate blood glucose levels, among other effects. And then the inner portion of the cortex, which is called the reticularis, produces androgens, which are similar in action to testosterone. They increase muscle mass and hematocrit and metabolic rate and bone density, among other effects. So it's really two organs in one. The pancreas is an exocrine and endocrine organ. The exocrine function is to produce digestive enzymes that help break down the macronutrients in our diets. So it produces enzymes that break down protein and fat and sugars. While the endocrine function is to produce two hormones that control our blood sugar levels, insulin and glucagon, both peptide hormones. Essentially, insulin is a hypoglycemic hormone, causes blood glucose levels to drop by inducing target tissues to take glucose out of the blood for fuel, or in the case of the liver, to take glucose out of the blood to convert it into fat or into glycogen. And glucagon has the opposite effect. Glucagon promotes the release of sugar into the bloodstream, so it promotes glycogenolysis. It also promotes gluconeogenesis in the liver, and the result is to elevate blood glucose levels. So these two hormones work together to try and keep blood sugar within a nice tight limit. And then the gonads, in the females those are the ovaries, and in the male those are the testes, are also exocrine and endocrine organs. The endocrine function of the testes is to produce testosterone and to a lesser extent estrogen and progesterone. The result is to control male secondary sex characteristics and also spermatogenesis, and in the female, the ovaries produce estrogen and progesterone and to a lesser extent testosterone, and that controls the ovarian and uterine cycle and the maintenance of female secondary sex characteristics. Hormones are chemical messengers that travel a great distance in the circulatory system and affect their target tissues, which are those cells that display a receptor for that particular hormone. They can also travel in the lymph to exert their effects. Autocrines are chemicals that exert their effect on the cell that produces them, while paracrines produce their effects on neighboring cells. Autocrines and paracrines are not technically considered part of the endocrine system, but the mechanisms are very similar. There are two main classes of hormones: amino acid-based hormones, which include amino acid derivatives such as catecholamines, as well as peptides and proteins. Most amino acid hormones are going to be polar, so they tend to be water-soluble with the exception of the catecholamines. Steroids are made from cholesterol and are nonpolar. Gonadal and adrenocortical hormones are based on the steroid nucleus, and their receptors, instead of existing on the surface of the cell membrane, are actually in the cytoplasm of the target tissue that they're going to exert their effect on. Although hormones circulate systemically, only those cells that have a receptor for that hormone will be affected by changes in those hormone levels. Target cells are defined as tissues that have receptors for that specific hormones. Hormones can alter a target cell's activity. It may be to change the plasma membrane permeability and or the membrane potential by opening or closing certain protein channels in the membrane. It may stimulate the synthesis of enzymes or proteins, and it can activate or deactivate enzymes, or induce secretory activity, or stimulate cell division. How do hormones exert their effects? Well, they can act at receptors in one of two ways, depending on the chemical nature of the hormone and the location of the receptor. All water-soluble hormones, which include all amino acid-based hormones except for the catecholamines, T3 and T4, made by the thyroid, act on plasma membrane receptors through a G protein second messenger and do not enter the cell, and that's due primarily to their large size and their polarity. Lipid-soluble hormones, such as the catecholamines and the steroid hormones, pass through the cell membrane, having receptors that exist in the cytoplasm. What's happening when the hormone passes through the membrane is it will find its cytoplasmic receptor, and that complex will then be escorted to the nucleus through a nuclear pore, where it will induce transcriptional activity from target genes, thus exerting its effect. They can enter the cell primarily because they are nonpolar, thus the cell membrane does not present a significant barrier to them going from the outside to the inside of the cell. What you're looking at here is the second messenger system employed by most hormones that are amino acid derivatives or peptide hormones. And they work via a second messenger called cyclic AMP through a G protein that's attached to the receptor, which has the ligand binding portion on the outside of the cell membrane. The first thing that happens is that the hormone, which we defined as the first messenger, binds to the receptor, which activates the G protein. The G protein binds GTP, and this will then in turn shuttle across the membrane to an enzyme embedded in the membrane called adenylate cyclase, which has as its enzymatic activity the production of cyclic AMP from ATP. The cyclic AMP will then act on downstream targets. It can activate protein kinases that phosphorylate proteins. It can also have other potential effects. The cyclic AMP signaling mechanism has the activated kinase phosphorylate other downstream targets, and this will activate certain proteins and inactivate other proteins. The cyclic AMP is broken down by the enzyme called phosphodiesterase, which is constitutively present in the cytoplasm. As a result, the intracellular enzymatic cascade can exert its effect rapidly because at each step in the cascade we amplify the signal. You have to remember that this diagram, while it shows you a one-to-one correspondence in each step in the chain, in reality what you do at each step is you can interact with more than one downstream target. So for instance, this activated receptor can activate more than one G protein, which can activate more than one adenylate cyclase, which can make thousands of molecules of cyclic AMP, which can exert their effects on thousands of molecules of inactive protein kinase, and so on. The result is that the signal gets very large very quickly as a result of geometric amplification. It also provides the ability to easily regulate the cascade by turning off the first and second steps in the cascade. We can quickly cut off the response because we know that resident in the cytoplasm are enzymes that will quickly break down the cyclic AMP. In addition to cutting off those first two steps, these combined effects will result in a cessation of the cellular response. And so we see cascades in a variety of places. This is an intracellular cascade. Extracellular cascades also exist. For instance, blood clotting is an example of an extracellular cascade. And the mechanism is very similar. By amplifying the signal, we make a huge biological response relatively quickly, and we also have the ability, as a result of being able to control the early steps in the cascade, of reversing the effect. Okay, there are other second messenger systems that we should be aware of. The PIP2 calcium signaling mechanism involves a G protein and membrane-bound effector, which is called phospholipase C, which splits a membrane phospholipid called PIP2 into two second messengers, diacylglycerol and inositol triphosphate. Diacylglycerol activates the protein kinase, which will in turn phosphorylate downstream proteins, while the inositol triphosphate induces calcium release. The calcium ions act as a second messenger, altering enzyme activity and opening or closing channels, or binding to the regulatory protein calmodulin. Calcium-bound calmodulin activates enzymes that amplify cellular response to the initial signal. And again, we see here another example of a perfect cascade. We have initial interaction between the ligand and the receptor. The receptor in turn activates phospholipase C, which can in turn produce massive amounts of DAG and IP3 from the PIP2, which is part of the structural makeup of the cell membrane, and then they will bifurcate, causing calcium release and activating protein kinases in order to amplify the signal and produce a profound cellular response. Other signaling mechanisms include cyclic GMP. Some hormones work without second messengers. As an example, insulin has a receptor that is a tyrosine kinase enzyme that autophosphorylates when the insulin attaches to it, and this serves as a docking station for downstream proteins that trigger the cell response. Here we're looking at the mechanism of steroid hormone activation of target tissue. Steroids are nonpolar hormones. As a result, they can very easily pass through the cell membrane because the nonpolar portion of the phospholipid bilayer does not represent a huge energy barrier to that movement. It can diffuse into the target cells, binding intracellular receptors. The complex can then move through a nuclear pore and attach to a receptor binding region on DNA upstream of target genes, inducing transcription. Those messenger RNAs can then leave via the nuclear pore, where ribosomes will attach to them and generate a novel protein product which will serve as the cellular response. So here we have direct gene activation as the mechanism. Target cells have to have specific receptors to bind the hormone in order to respond to changes in that hormone's level. Adrenocorticotropin hormone receptors are found only on the cells of the adrenal cortex, while thyroxine receptors are found on nearly all cells of the body. Thus, this defines the scope of the target. ACTH will only induce action in the adrenal cortex, while thyroxine will hit every tissue in the body, generating its effect. Target cell activation depends on blood levels of the hormone, relative amount of receptor on the target cell, and the affinity of binding between the receptor and the hormone. Hormones influence the number of receptors on their targets. In upregulation, target cells form more receptors in response to low hormone levels, while in downregulation, target cells lose receptors in response to high hormone levels. So you could think of upregulation as the target tissue reaching out to try and find its chemical signal in order to produce the appropriate activity. In downregulation, you could envision that as swamping the system. By overloading it with too much hormone, the targets become less sensitive, and the result is that the signaling doesn't work as well. An example of that is type 2 diabetes, where as a result of extensive insulin production over a lifetime of consuming high-calorie foods, we end up with target tissues that are not as sensitive to insulin levels as they otherwise would be, and the result is that we have a difficult time regulating our blood sugar. How do we control hormone release? Well, there's a variety of methods. Usually negative feedback is part of this mechanism. We want to keep hormone levels within a tight range to maintain homeostasis. The endocrine gland stimulated to respond to the signal that will either induce hormone release or keep hormone from being released include humoral stimuli, which are changes in chemical levels in the blood, neural stimulus, where the nervous system actually induces the target endocrine organ to secrete, and hormonal stimuli, where levels of hormone in the blood or in the lymph trigger the production of a chemical command that will induce the release of that hormone. So here are some examples. This is parathyroid hormone. What you're looking at here is an example of humoral stimulus. Changing blood levels of ions and nutrients directly stimulate the secretion of this hormone. Here the trigger is calcium levels. Dropping calcium levels stimulate parathyroid glands to generate parathyroid hormone. The result is the elevated parathyroid hormone levels increase the amount of calcium in the blood. The result then feeds back to the original organ, and parathyroid hormone goes back down, hence negative feedback. In neural stimulation, nerve fibers stimulate hormone release. As an example, we have the adrenal medulla, which is innervated by the preganglionic neurons of the sympathetic division of the autonomic nervous system. The result is to secrete catecholamines directly into the bloodstream and thus exert their effect on target tissues. Hormonal stimuli rely on hormone levels in the blood or the lymph in order to induce the desired endocrine gland to generate its command. As an example, hypothalamic hormones stimulate the release of most anterior pituitary hormones, which stimulate their targets to secrete their hormones. The hypothalamic pituitary target endocrine organ feedback loop involves hormones from the final target organ inhibiting the release of hormones from the anterior pituitary and in many cases also the hypothalamus. So if we take one example and look at the thyroid gland, we see that dropping levels of thyroid hormone will induce the secretion of thyrotropin releasing hormone from the pituitary or from the hypothalamus. This will flow in the bloodstream through the hypothalamic hypophyseal portal system to the anterior pituitary and cause the anterior pituitary to produce thyroid stimulating hormone, which in turn will cause the thyroid gland to produce its components, thyroxine and T3. As the levels of thyroxine and T3 rise, it feeds back to the anterior pituitary and the hypothalamus, reducing production of TRH and TSH, and thus the negative feedback loop completes. Until, obviously, the levels of thyroid hormone fall again. How does the nervous system operate to modulate hormone levels? Well, it modifies the stimulation of endocrine glands and their negative feedback mechanisms. An example is under severe stress, the hypothalamus and the sympathetic nervous system are activated. The result is that blood glucose levels rise. The nervous system can override normal endocrine control in many cases. Hormones circulate in the blood as either free or bound hormone. Steroids and thyroid hormone are attached to plasma proteins, and the reason for this is that they're nonpolar. As a result, they need a vehicle to carry them in the plasma because they don't dissolve well in blood plasma. All other hormones circulate without carriers. Since they're polar, they dissolve well in plasma. The concentration of circulating hormone will affect the rate of hormone release, the speed of inactivation and removal from the body. Hormones are removed from the blood by enzymes that break them down, the action of the kidneys, the action of the liver, which has a role in the half-life of that hormone, which is defined as the time required for the hormone's blood level to decrease by half, which will vary from fractions of a minute to a week. So these systems frequently work together. The degrading enzymes can set up the liver to couple hormone that needs to be cleared to groups that dissolve very easily in water. Then when that blood gets to the kidneys are able to secrete the degraded hormone in the urine stream. And this results eventually in the levels of the hormone in the blood dropping. So if we're looking at hormone effects on target tissues, what are the scale of these effects? Some responses are immediate. Some, especially steroid hormones, last from hours to days, and some have to be activated in target tissue. The duration of hormone activity can be limited, ranging from 10 seconds to several hours. The effects can disappear as soon as the blood levels drop. Some persist at low levels. We also observe that multiple hormones can act on the same target at the same time. This can produce effects such as permissiveness, where one hormone can't exert its effect without the other hormone being present, or synergism, where the combined action of two hormones together produces a greater effect on the target than either effect of each hormone individually, and antagonism, where one or more hormones oppose the action of the other hormone. An example of antagonism, for instance, would be the action of parathyroid hormone calcitonin. One acts to elevate blood calcium levels, and one acts to reduce blood calcium levels. An example of synergism could be the action of epinephrine on target tissues in combination with the action of cortisone and cortisol on target tissues. The cortisone and the cortisol act to elevate blood glucose levels. The action of epinephrine is to elevate metabolism. The pituitary gland has two major lobes, the posterior pituitary and the anterior pituitary. The posterior pituitary is strictly neural tissue, while the anterior pituitary is glandular tissue. So we call the posterior pituitary the neurohypophysis and the anterior pituitary the adenohypophysis as a way of designating the two different tissue types. Here we're looking at the pituitary gland as it dangles from the infundibulum under the hypothalamus. The posterior pituitary is a downgrowth of hypothalamic neural tissue and has a direct nerve connection to the hypothalamus through the hypothalamic hypophyseal tract. The nuclei of the hypothalamus synthesize neurohormones such as oxytocin and ADH, which are then transported to and stored in the posterior pituitary until they need to be released into the bloodstream. The anterior lobe is under the governance of the hypothalamus via inhibiting and releasing factors that travel in the hypophyseal hypothalamic portal system to the anterior pituitary, exerting their effects. The anterior lobe originates as an outpouching of the oral mucosa. It has a vascular connection to the hypothalamus. The hypophyseal portal system is that connection. Primary capillary plexes are found in the hypothalamus. The hypophyseal portal vein carries the blood to the secondary capillary plexus, which basically encloses the entire anterior lobe, and this carries the releasing and inhibiting hormones to the anterior pituitary that regulate its release. Oxytocin and ADH are the posterior pituitary and hypothalamic hormones. Oxytocin is going to have as its primary mode of action the inducement of smooth muscle contraction, while ADH causes the body to retain fluids. Each is composed of 9 amino acids, and they're virtually identical, differing in only 2 amino acid portions of the primary sequence. Oxytocin is a strong stimulant of uterine contraction and is released during childbirth. It's also a hormonal trigger for milk ejection and acts as a neurotransmitter in the brain, so it has a dual role. While vasopressin inhibits or prevents urine formation, thus regulating water balance, it targets the kidney tubules to reabsorb more water. Its release is triggered by pain, low blood pressure, and certain drugs. It's inhibited by alcohol and diuretics, and this is one of the reasons why when you drink a lot, you urinate a lot, and this is the trigger for the physiological response known as a hangover. At high concentrations, it promotes vasoconstriction. Diabetes insipidus is an ADH deficiency due to damage in the hypothalamus or the posterior pituitary. The result is the individual has to be well hydrated because they have consistent water loss. It's a condition known as polydipsia, they're thirsty all the time. The condition of increased urination is called polyuria. The syndrome of inappropriate ADH secretion causes the body to retain more fluid. This can result in headache and disorientation. The treatment is fluid restriction and blood sodium level monitoring. Other anterior pituitary hormones include human growth hormone, thyroid stimulating hormone, adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, and prolactin. All of these are proteins. All except growth hormone activate cyclic AMP second messenger systems on their targets. TSH, ACTH, FSH, and LH are trophic hormones because they regulate the secretory action of other endocrine organs. Growth hormone is made by somatotropic cells that direct actions of metabolism. It increases blood levels of fatty acids, it encourages the use of fatty acids for fuel, and it encourages protein synthesis, so it's an anabolic hormone. It decreases the rate of glucose uptake and metabolism, thus conserving glucose for the nervous system. Glycogen breakdown and glucose release is triggered. Their increased levels in the blood will have an anti-insulin effect. Indirect actions on growth hormone on growth include the mediation via growth-promoting proteins called insulin-like growth factors, which stimulate the uptake of nutrients, which leads to DNA and protein synthesis, the formation of collagen, and the deposition of bone matrix. Major targets include bone and skeletal muscle. Growth hormone release is regulated by hypothalamic hormones. Growth hormone releasing hormone stimulates the release of growth hormone, while growth hormone inhibiting hormone, known as somatostatin, will inhibit the release of growth hormone. Ghrelin, which is a hunger hormone, also stimulates the release of growth hormone. What you're looking at here is just a chart that shows you how the release of growth hormone is regulated. So again, because we're talking about the anterior pituitary, the hypothalamus can secrete growth hormone releasing hormone and growth hormone inhibiting hormone to regulate GH production by the anterior pituitary. So let's start with the stimulatory wing of this whole process. We have feedback that can promote the release of growth hormone. Okay, examples could be reduced levels of growth hormone in the blood. Okay, as one example. Growth hormone releasing hormone stimulates the anterior pituitary to generate growth hormone. The direct effect is to target liver and other tissues to produce insulin-like growth factors that will cause an increase in the production of skeletal and cartilage tissue, and also stimulate increased cell growth and proliferation in tissues outside the skeletal system. Indirect effects encompass most of these actions. Direct effect includes an increase in fat breakdown and carbohydrate breakdown. The result is that the fat that's released into the bloodstream is used by the tissues that are in anabolic phase to generate energy. Carbohydrate metabolism is directed towards keeping the nervous system functioning, while most of the other tissues use the fat as a primary fuel source. What happens as the levels of insulin-like growth factors and growth hormone itself increase is that we will eventually feed back to the hypothalamus and the anterior pituitary and cut off production of growth hormone. In hypersecretion, prior to the end of puberty, gigantism results. In adults, hypersecretion after puberty results in a condition known as acromegaly. To see an example of an individual that had both of these endocrine disorders, if you look up or Google Andre the Giant, you'll see somebody who suffered both from gigantism and acromegaly. In hyposecretion, we see pituitary dwarfism, where the body proportions remain normal, but the overall stature is diminished. Thyroid stimulating hormone, as the name would imply, stimulates the thyroid gland to produce T3 and T4. It's produced by the thyrotropic cells of the anterior pituitary and stimulates normal development and secretory activity of the thyroid gland. The release is triggered by thyrotropin releasing hormone from the hypothalamus, and it's inhibited by elevated blood levels of thyroxine that act on the pituitary and the hypothalamus. So again, we see an example of hormonal regulation at the level of the hypothalamus and the pituitary gland. Adrenocorticotrophic hormone is secreted by the corticotrophic cells of the anterior pituitary. It stimulates the adrenal cortex to release corticosteroids. Regulation of ACTH production is triggered by hypothalamic corticotropin releasing hormone, usually in a daily rhythm. Internal and external factors such as fever, hypoglycemia, and stressors can alter the release of corticotropin releasing hormone. Gonadotropins, as the name implies, target the gonads. In the females, those are the ovaries, and in the males, those are the testes. The two gonadotropins are follicle-stimulating and luteinizing hormone, which derive their names from their targets in the female reproductive system. Follicle-stimulating hormone targeting the follicles of the ovary, which contain the eggs, and luteinizing hormone targeting the follicle after the egg has been released in the process of ovulation in order to stimulate the formation of the corpus luteum and also induce the corpus luteum to secrete its hormones, estrogen and progesterone. It's secreted by the gonadotropic cells of the anterior pituitary. FSH stimulates gamete production. In males, that's spermatogenesis, and in females, that's oogenesis. While luteinizing hormone stimulates the production of gonadal hormones such as estrogen, testosterone, and progesterone. It's absent from the blood in prepubertal boys and girls under normal circumstances. Its release is regulated by gonadotropin releasing hormone during and after puberty, and it's suppressed by gonadal hormones via feedback. Prolactin is secreted by the prolactin cells of the anterior pituitary, and it stimulates milk production. Its role in males is not well characterized. Its release is regulated by prolactin inhibiting hormone. Blood levels tend to rise towards the end of pregnancy, and this stimulates milk production from the breast tissue. Suckling also stimulates prolactin release and promotes continued milk production, while hypersecretion causes inappropriate lactation, lack of menses, and infertility in females and impotence in males. We should also add that the hormone oxytocin plays a role in milk production as well, as it induces milk release. Okay, so the prolactin is milk production by the breast tissue, and oxytocin stimulates the myoepithelial cells in the breast to actually squeeze the milk into the lactiferous sinuses and inject it through the nipple so that the baby can get the nourishment. Okay, that brings this portion of Chapter 16 to an end. I will join everybody in the podcast on Chapter 16, Part B. So see you soon.